Hi, everybody. I'm Cristina Zamora. I'm doing the PhD here in the group of Forest Pathology in the University of Valladolid. The topic of my presentation this year is genome-wide identification and characterization of defense-related long non-coding RNAs in non-model plant Pinus radiata, quite long. And now you are wondering what the hell is a long non-coding RNA. So let's get started. As all you know, the DNA is made up of genes that, according to the general dogma of molecular biology, they're transcribed in messenger RNA and then translated to proteins. So we can say that the DNA is encoded for proteins. However, only 1.5 of the transcribed RNA is translated to proteins. So this implicates RNA to have more roles or diverse roles others than being just an intermediated component in the genetic flow. Okay, the non-coding part of the genome can be categorized by structural non-coding RNAs, which are the ribosomal RNA or transfer RNA, these well-known RNAs that are implicated in the genetic flow, and the regulatory non-coding RNAs, where we have the small non-coding RNAs of more than 200 nucleotides that are well known, studied in eukaryotes as regulatory molecules. And more recently, with the development of advances sequencing technologies and computational methods, a new class of non-coding RNAs has emerged. Here, all the transcripts of longer than 200 nucleotides without coding potential have been included. This class or these transcripts have been ignored during these last years, but now with the accumulation of evidence, we can say that they have important roles in the transcriptional system. The long non-coding RNAs can be divided according to their genomic location and orientation. They can be transcribed in sense or antisense with respect to the coding genes, and they can be between genes like intergenic long non-coding RNAs, or into the intron of the coding genes like intronic long non-coding RNAs. They have a lot of mechanisms of action, like molecular signaling or precursor of microRNAs and so on. They have a lot of functions and mechanisms. Functionally, in general, long non-coding RNAs can act in cis by regulating nearby genes, the genes close to them, or they can function in trans by regulating expression of distant genes in other parts of the genome or far away from them. Thousands of long non-coding RNAs have been identified in model plants such as Arabidopsis and so on. However, no reports investigating long non-coding RNAs in conifer trees under the challenge of fungal infection had been published so far. Indeed, we have published for the first time the identification and characterization of long non-coding RNAs in Pinus radiata in response to an infection of Fusarium circinatum. Okay, this is the forest disease in which Fusarium circinatum is involved, the pine pitch canker. For that, we inoculated Pinus radiata seedlings with the pathogen, with Fusarium circinatum, and 4 days after the inoculation, we extract all the RNA from the tissue in the point of inoculation, was extracted, and then sent to sequence, massively sequence. The small fragments or reads from the sequencing were suggested to a long bioinformatics pipeline that we are not going to see in detail because we have not time enough. However, I want to stress some important steps, such as the assembly of the short sequence reads in order to generate all the transcripts of the pine, and then the application of different filters to identify the long non-coding RNAs among all the transcripts of the pine. In addition, in order to identify the long non-coding RNAs involved in the response to the pathogen, we perform a differential expression analysis between the inoculated and the mock inoculated or the control plants. In addition, the genes predicted to be regulated by these differential expressive long non-coding RNAs were analyzed in order to find, the function in which the long non-coding can be involved. So, moving to the results, we could identify 13,312 long non-coding RNAs in the pine, the majority being intergenic long non-coding, the ones that are located between genes. Compared with the protein-coding RNAs, compared with the normal genes, these long non-coding RNAs were shorter, had lower expression, lower guanine cytosine content, and had fewer and shorter exons. This was consistent with the long non-coding identified in other organisms in the world, like plants, animals, or humans. The differential expression analysis identified 174 differential expressive long non-coding RNAs in response to the pathogen. These long non-coding were predicted to be regulating 39 normal genes because they were found in a genomic window of 10 kilobases upstreams, 100 kilobases downstreams from these long non-coding RNAs. And the functional prediction of these differential expressive long non-codings was based on the functional annotation of these these nearby genes. These functions were enriched to several defense mechanisms, such as cell wall modification, response to stress, or regulation of phytohormones. On the other hand, the identification of trans-acting long non-coding RNAs was based on the co-expression of genes and long non-coding RNAs. Two models of co-expression were identified in the analysis. The genes and long non-coding RNAs included in these models may act together or may be similarly regulated in the defense responses of the plant. The largest model was enriched. The functions of the largest model was enriched mainly for pectin esterase activity and cell wall remodeling. So we can say our results suggest that long non-coding RNAs are involved in the cell wall remodeling and lignification process as defense mechanism, which had been previously reported in other plants such as cotton, populus, or paulonia. So So our analysis can confirm that the pine-long non-coding RNAs are involved in the defense of biotic stresses, as occur in other organisms such as other plants, animals, and even humans. And thank you so much for your attention.